ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. What's good with everybody? How y'all doing, man? ETM Hotel, Rennie Sean. Welcome in peace. My name is Sean. I'd like to thank everybody for tuning in to Science with Sean. I am streaming from the Science with Sean channel, YouTube channel, and the Mossy Warrior Clan today. I see Nunu in the building. Omar, Donnie, Thurman, Teddy was good. There's some other people that may be listening or tuned in. Keenan was good. I haven't shared the show. I'm actually not going to share the show. I'm just going to jump straight into the information in just a second. I am still promoting the Science with Sean channel, trying to build up the audience. Again, you should be able to locate and find the Science with Sean channel. Let me see. I might not even be an admin. Nope. I'm not an admin on my own. What up, Trick? What's good? But yeah, man, I'm gonna put the link in the chat so that people could I'm gonna put the link. Oh man. People can subscribe to the channel. You know? So it's probably gonna go on both. Make sure you like the show, share the show, tell a friend, tell a friend. I'm not gonna share it. I know people get tired of me dropping stuff in the inbox, so I'm trying to refrain from putting information in people's inbox and then see if they'll naturally gravitate to it. I guess I can make it sexy, you know, have some people on here and be confrontational and talk about religion for 8 hours or something like that. And maybe they'll come in, or if I speak their language, you know. I was about to kind of shut down and probably not present again for quite some time. So I am working on this other book. I got one of the 2 books done, and I got my chapter for volume 4 done. So I'm about 60% where I need to be as far as my writing material for this year. I say 60 because as soon as I finish this other book, I think Ben's going to drop a book, and hopefully Nahisi drops his, and then the journal will be up next. Man, we talking about variants, and I see that we got somebody in the chat. Tiarka asked what made the Mossy a warrior clan. Well, first, the Mossy is not a warrior clan. If you read volume 1 of Spears of the Mossy, Black Panther and Brother Ankh lay out the foundation of how the Mossy warrior clan was formulated. But as far as the Mossy go, the Mossy rejected Islam and Christianity for quite some time. And they held their own against people who were trying to ransack, you know, the areas and things of that nature. And they did a lot of ransacking. If you look back to the days of even Askia Muhammad, but before Askia Muhammad, Sonny Ali, there was a lot of warring going on between the Masi and you know, and Son and them. They were just going back and forth. However, you know, that little whole Islam thing finally got an opportunity to dominate the way and dominate the trade and dominate in other areas. Therefore, kind of go from there. Matrilineal Fulani. Well, Sonny Ali hated the he he hated the Fulani. He was beheading the Fulani and waging war on the Fulani like it wasn't nothing. Expanding the Songhai Empire. Yeah. So yeah, you know, hey, the Fulani got some good in it. One thing about the Fulani, if you're unaware of it, but we have a lot of things on this channel. We have a few things on this channel that are related to the Fulani culture, culture wise. There's a few things that we dropped on here, but I can't see the Fulani man in you. You know, the Fulani women have opportunities to yeah, it's because of they ties to Islam and a whole bunch of other stuff that the Fulani was doing. But the women, you know what I mean? If you know, they got this thing called Mr. Steal Your Girl, right? So you got a girl in Fulani culture. If she sees somebody else that might seem to make her more womanly than you, they got this thing with a dude to try to steal your girl, sneak off with it and all of that, the whole nine. But you know, it's some cultural aspects on here, man, regarding Fulani. The men dress up. They think they the prettiest men on earth. They dress up, and they put all kind of, you know what I mean, stuff on, you know, tribal markings and paint markings on their face to make themselves look brighter. And then they stand in line next to one another, and yeah, they stand in line and they dance and they try to stand out to the women. They make certain noises and things like that. And you know, to get a certain female's attention. Yeah, that's part of the food. Yeah, the Wudabi. That is the Wudabi. They're Fulani, Zane. You seen my Facebook post. That's probably why you threw out the Wudabi like it. I got that post on Facebook about the Fulani. Uh-huh. The Wudabi it is. But yeah, Taraka, that's you know how the name circulated, and that's a little bit about the Fulani. There's more. Don't think that they were just weak because the Songhai Empire spread and dominated the Fulani people. That wasn't the case. The Fulani had some nice battles. I'm excited about chapter 4. I'll tell y'all that now. It's different. It's a different perspective than now that I went. Well, not really. It's still on West Africa, but what I did for chapter 4, people will appreciate. Um, you know what I mean? Well, I don't know if people appreciate it or not, but it'll put it in context. Uh, Not too many people are familiar with what's going on with that, so it's kind of, you know, that kind of dictate the terms and whatnot of what people can learn. So and then after that I have written, I hope people are prepared for that. Like I said, um, you know, I'm how many pages am I in on this one? I am 53 pages in on this one, and I am at the end of a chapter. So I just gotta close out that chapter with some stuff that I need to add in it. And then I'll be on chapter 4 of that book, beginning chapter 4. And I'm gonna make this book digestible and relative, but most importantly, an opportunity for people to learn more. And maybe it inspire you to go behind me and, do some further studying. Yeah, the Wudabi people of the Fulani, they are more recognized because their culture is commercialized. So you will see what up here. So you will see the Fulani, well, you'll see the Wudabi images all over Google, Chrome. Um, People are fascinated with the Wudabi. You know what I mean? Um, there's some other intricate pieces in the Wudabi that are familiar with what's going on. But yeah, on the Masa Warrior clan, we do have some information, man. And it's not long presentations or anything like that. We got some quick stuff too, that you can learn from. So if you're not subscribed to the channel, subscribe. Again, I am streaming from Science with Sean and the Masa Warrior clan. I'm gonna go ahead and get going today, because we're gonna have plenty of time to get back to West African culture and still be able to maintain balance with Science with Sean, because I want to kind of expand beyond just what people think I know, just vaccines. So yeah, let me share my screen, and I'm gonna keep this up for both channels. I prefer really everybody to kind of get to, get all them tabs up there, cross-referencing and blah, 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 and looking up stuff. Yeah, man, you got to go in. So what the variant? Let's go. By you know who. Copyright disclaimer under Section 107 of the Copyright Act 1976. Allowance is made for fair use for purposes such as criticism, comment, news reporting, teaching, scholarship, education, and research. Fair use is a use permitted by copyright statute that might otherwise be infringing. Today, we're going to talk about, like I said, variants. So we'll get into the introduction, hypothesis, research question, definitions, conclusion, core sources. In this short lecture, our focus is to understand virus variants and should we take them seriously. According to the media outlets, a lot of emphasis has been put on variants, confusing them at times with strands. I believe that the pandemic exposed our ignorance as a people, and it put humanity on notice by crippling economies and families until this day. When news cycles began to share stories about viruses with headlines of a new strand existing, we have no idea if it's a serious matter or not. Some people may be under the impression that strands are variants and vice versa. The problem exists because of our inability to actually rely on studying virology and getting familiar with the scientific terms. We'll take a look at a few definitions. A virus variant is an isolate whose genome sequence differs from that of a reference virus. No inference is made about whether the change in the genome sequence causes any change in the phenotype of the virus. I want to stay here real quick and kind of help unpack this very first two sentences in the first definition. An isolate whose genome sequence differs from that of a reference virus. The genome. This is how unique genetics is when it comes to understanding viruses and virus variants. Now, I might sound a little geeky or nerdy right now, but just researching, reading, and learning about viruses and things of that nature, and putting my my book together and stuff, you start to realize that something that Vincent Racanelli says, that uh, in order to understand viruses, you have to be, what do you call it? He it's a it's a intra. No, he didn't use the word intracellular. He didn't use that word. He used a different word, and I can't think of it at the moment. But it's uh it's a multidisciplinary field or study. Understanding viruses is a multidisciplinary study. Why? Because it deals with genetic sequences. It deals with evolution or natural selection, because you got mutation and adaptation. It deals with microbiology. Um, you know, because you're talking about cells and things like that. So it's a multidisciplinary study. And just knowing a little bit in each one of those areas can help you understand viruses. So, uh, and then the second line, it says no inference is made about whether the change in the genome sequence causes any change in the phenotype of a virus. How many of you and people in the chat should know this, but how many people knew that a virus had a phenotype? When you hear the word phenotype in other discussions, you're usually talking about skin color, right? You're differentiating basically a light-skinned person from a fair-skinned person, from a darker complected person, from a uh, mixed person, phenotypically speaking. So you're talking about different shades of white, black, brown, dark skin, light skin, complexions, right? But anyway, viruses also have a phenotype. There you go. Take that with you. Absolutely, EIE. hmm Now, the meaning of variant has become clouded in the area of a whole viral genome sequencing, because nearly every isolate may have a slightly different genome sequence. Now, this other definition says the mutant forms of viruses are called variants. Just keep it simple right there. In some cases, the variants are genetically distinguishable, my bad, but not different. Their mutations have no effect on their biology. And then the next definition says a virus with one or more mutations is referred to as a variant. Most of the time, variants do not affect a virus' physical structure, and in those instances, they eventually disappear. However, there are certain cases when a mutation impacts part of a virus' genetic makeup that does change its behavior. When Charles Darwin first wrote about the law of natural selection, he imagined that the evolution in response to selection would be slow. Humans would be left, he thought, to study the consequences of natural selection, but unable to see the process in real time. It would have delighted Darwin to know that in this way that he was wrong. The good news is that the consequences of natural selection, evolution, and even the origins of species can be observed in real time. This is true, for example, with experiments done on bacteria in laboratories. It is also true with regard to the virus that causes COVID-19. Much as the finches, now called Darwin's finches, evolved after having arrived in the Galapagos Island, the virus that causes COVID-19 is evolving rapidly as the virus continues its global spread. In this light, I thought it would be worth sitting down virtually, of course, with a group of ecologists and evolutionary biologists who have focused their life work on viruses and the peculiar ways they change. Natural selection and adaptation may play a role in determining which variants or strands become dominant, as in those that develop competitive advantages, such as increased transmissibility or the ability to evade immunity. Many virology terms are being used these days by people who do not understand their meaning. Included are journalists, medical doctors, scientists, lawyers, and people from all walks of life. In the normal times, this word misusage would be so rare that it would not matter. However, because we are in a viral pandemic that affects nearly everyone, I will attempt to explain the meaning of the virus, isolates, variants, and strands. Many of the terms used in virology are ill-defined. They have no universally accepted definitions, and there is no Bible with the correct meaning. As each of us are trained by other virologists, we hear them using terms in certain contexts and we copy their usage, whether or not it is correct. I learned many good things from my mentors, but also many things that are wrong. I kept that because, I look at Racanelli as not only an inspiration, but a teacher. Now, an important piece of information to know from the start relates to sex. In many species, sexual reproduction allows the genes from two or more fungi or complex parents to come together to yield offspring that are genetically novel and also have novel physical traits. Viruses are different. They can sweep genes in wondrous ways, but the far more common source of the variation results from mutations, chance changes in their genetic letters of their genes. Of course, our genes mutate too, but viruses mutate at a rate much higher than in cellular life forms, such that mutations rather than the reshuffling of genes among offspring creates most of the genetic variations natural selection can act on in a viral population. Mutant forms of viruses are called variants. In some cases, the variants are genetically distinguishable but not different. Their mutations have no effect on their biology. In other cases, the variants are genetically distinguishable and differ in their biology. Those variants are called strands. This is a subtle distinction, but an important one. I'm going to read that again to you, okay? In some cases, the variants are genetically distinguishable but not different. Their mutations have no effect on their biology. Now, in other cases, the variants are genetically distinguishable and different in their biology. When they are distinguishable and different in their biology, they are called strands. When there's no change, no difference, the mutation has no effect on the biology, they're called variants. So now you know the difference between variants and strands. Major consequences of variants include CDC 2021A, increased transmissibility and ability to spread faster. That's one consequence. Altered disease severity, either milder or more serious. It's another consequence. Lower detection by viral diagnostic tests, however, most reverse transcriptase or polymerase chain reaction tests utilize a variety of targets that can detect the virus despite the presence of mutations. Reduced response to treatments and medications currently used to treat COVID-19. Those are consequences. Improved ability to evade natural or vaccine-induced immunity is another. This is why they were in such a hurry to try to vaccinate so many people in the population, is because if new variants emerge or will emerge or already have emerged, that, they will have the ability to, evade the vaccine-induced immunity and causing roughly, more breakthroughs. But at this moment, we're not seeing that many. All right, so here's variants of concern. You have the UK variant, which we talked about during the whole evolution of the very first coronavirus variant that was sequenced. But the UK variant is B116. It now has a new name, right? Because they're using those alpha, beta, all those names now. and we'll get into that maybe. The South African variant, which is the B1351. The Brazil is P1. They simplified it. And then you have two California variants. You have the B1427 and the B1429. Each one of these has a more lets you know there's 50% more transmissible, which means if you come into contact with, it's a possibility if you're unvaccinated that you could be reinfected by a different variant that's more transmissible, likely to cause more severe diseases, and if you got vaccine, then you already know if you're good to go or not. All right. Now, the California variant is just 20% more transmissible, where the UK and South African variant are 50% more. And then you talk about, like the California variant may cause more severe disease, but more research is needed because it's new. It's novel in the area as far as the variant is concerned. And that's because it mutated. It adapted to California lifestyle. Now, WHO, which is the World Health Organization SARS-CoV-2 variant statement, said has received several reports of unusual public health events possibly due to variants of SARS-CoV-2. WHO routinely assesses its variants of SARS-CoV-2 result in change in transmissibility, clinical presentation, and severity, or if they impact on countermeasures, including diagnostic therapeutics and vaccines. Again, a look at the coronavirus variants of concern. And when they became available, you see the B117 first recorded in September 2020. Its destination, again, was the United Kingdom, UK. The key mutations was in the spike protein. And then the N501Y, which is all three variants, have mutations in the receptor binding domain of the spike protein. The South African variant was first recorded in October, a month later. In December, a few months later, 2 months later in Brazil, the P1 mutation was located, and we seen the mutations from B1351 and P1. And then, you have the variant. The next one is the India variant, which was not mentioned in the last one, but of course, the Indian one was also detected in October. Many people are seeing both the UK strain, the UK variant and the Indian variant in India, and they could be developing a third potential variant that may be way more severe, way more transmissible, and the risk of disease could increase, and the severity is there if that's possible. The main concerns, like I said, with all of these viruses are always transmissibility, but the first two, which is the UK and South African variants, they are on those heavy because the Pfizer vaccine did not do so well against the South African variant. Its efficacy rate was reduced. Moderna did okay, did good, but both of them still remained high, and they still are trying to improve in those areas regarding the South African variant. I'm gonna be truthful. Let's move on. So the different variants, and you heard me mention them again, but I'm gonna be more specific here. Of course, the UK variant is first, which is the B117. This variant first identified in the UK, B1351, South Africa. G1 variant, A variant, that is originated with really supposed to be P1 in Brazil, originated in Brazil, but reported in Japan. And it was identified on 4 people screened upon arrival at the airport in Tokyo. There's one for California, which is the B1427 and the B129. Then you have the variants in New York. Yep, there's in Washington Heights and Manhattan. There's variants in New York, which is the B1526, B1525, and B1526. All right, so there's again, but the world was noticing the three major coronavirus variants, and the first one, of course, is the UK. I don't know if you actually can see my screen, and I can't zoom in on it, so I apologize. But the UK's variant is more contagious versus the other ones. Okay. Experts suspect the first case of the B117 originated in Kent, an English country in southeast London. From there, the variant quickly spread across the globe, with cases reported across Europe, Asia, and North America. Again, like we seen from the other one, it was 50% more easily and quickly transmissible. It was 35% more deadly. Where it says, and now it's detected in 94 countries and 46 states. And of course, we all know the current vaccine works well against this new variant. The B117, which is the UK variant, is a super spreader. The variant is primarily responsible for the rise in several hotspots in New York, Michigan, Wisconsin, and elsewhere. The B117 tends to dominate other variants in the community, has found to roughly double every 7 to 10 days, and is now about 26% cases in the nation. Good news, the vaccines are effective against it. Bad news, it's more infectious and can cause worse complications or death. Plus, only about 150 million doses of COVID-19 vaccines have been given in the U.S. so far. All right. Again, this one is in regards to the three major variants. This is the B1351 discovered in South Africa. Okay. It spreads more quickly, easily, and rapidly. It's detected in 48 countries. There is a possible reduction of vaccine effectiveness. It's better at evading our immune systems. In December, the UK reported 2 people who had recently traveled to South Africa were infected with the new variant. By the end of the month, the strands was found in Switzerland, Australia, and Japan, as well as in all cases linked back to South Africa. The California variants are about 20% more transmissible than the original SARS-CoV-2, but the vaccines are still fantastic and doing a really good job, says Stacy Wyman, a genomics researcher who leads the sequencing efforts at Berkeley Innovative Genomics Institute. South Africa and Brazil appear to render the current vaccine slightly less effective in experimental data. The B135, one is also about 50% more transmissible, but the rate for the P1 is not yet known. The B117 seems to be edging out the B135 and the P1, at least for now, although they are concerning. They are not playing a big role in epidemiology right now. Caitlin Rivers, senior scholars at Johns Hopkins Center of Health, says. All right, the next one is the P1 discovered in Brazil, spreads more easily and quickly, now detected in 26 countries, possible reduction in vaccine effectiveness, and better at evading the immune system. It was discovered in January 2021. 4 cases of the P1 variant were detected in Japan. The infected travelers had arrived in Brazil the same month. Cases of the P1 variant were reported in Italy, Germany, and the US. Here we have a daily infectious chart on the average. Source again is Johns Hopkins. As you can see, cases of variants begin to increase around December, and they spike in January, and then they begin to even its way off. And since the vaccine rollout, they are facing a decline in new infections across the country. In South Africa, that same case, that average day between June really late April, May, going into August, you've seen a rise there. But as of going into the new year, you've seen a rise and then a decline, showing that the South African variant, as it gets to new places, spikes and then, kind of evens out. The Brazil variant is staggering. It's up and down, up and down, up and down. It's consistently, doing what it's doing. Cases variants begin in January. They increase around, at the beginning of 2021, everything increased. Why? Because you had people gathering. You had Thanksgiving and Christmas. And people couldn't say no to their family members for Thanksgiving and Christmas. And after both of those holidays, going into the new year, we've seen an emergence in COVID-19 case increase.
Do you know what a virus variant is? Do you know what an isolate or a strain is? If you don't and you'd like to find out, keep on watching. I'm Vincent Racaniello, and I've been a virologist for nearly 40 years. And today I want to explain to you the terms virus isolate, variant, strain, and a couple more. As you're well aware, this is March 1st, 2021. Pandemic has been on for about a year now, caused by SARS-CoV-2. Many people are thinking about viruses, and so they're using terms that they may not be familiar with, like isolate, variant, and strain. So I thought I would tell you what I think those terms mean. Now, the problem is, as shown in this first slide, there's no universally accepted definition for these terms, strain, variant, and isolate, in the virology community. So how can we expect that everyone else gets them right? What happens is you're trained in someone's laboratory and you hear them say things and you associate a meaning with them, and that's what you do. And they may or may not be right. By the way, this little nugget of wisdom comes from this paper where Jens Kuhn is the first author. It was published in Archives of Virology in 2013. And I will make a few other quotations from that article as well. Now, despite this looseness of definitions, for example, there's no one place where you could go and find the definition for variant, for example, or isolate or strain. I looked in several textbooks. In fact, not even my textbook, Principles of Virology, co-authored with 4 other virologists. Not even there will you find the definition of these terms. I think we need to put it in the next edition, though. Let's go through and try and define them and put them in some kind of context with the current outbreak. Let's start with isolate. What is an isolate? An isolate is a virus that we have isolated from an infected host, and we have propagated that in culture. Now, isolating encapsulates the meaning. It means that you take a sample from someone who you think might have a virus infection, you put that sample in cells in culture, which is how we grow viruses, and you grow a virus, and now you have an isolate. Let me illustrate that for you here. So we do a nasopharyngeal swab for, we suspect someone might have COVID-19. You take the swab, you put it in a tube, which has a little fluid in the bottom. It's brought to a laboratory. And in the laboratory, you take a little of that fluid and you add it to cells that are growing in a culture dish. That is how we isolate viruses. And if that person had a virus in him or her, it would grow up in culture, and then you could take the medium from these cells and store them in little tubes, which is what we do in the laboratory. So that's what we mean by isolate. We have actually isolated the virus. Now, most of the time, we take this nasopharyngeal swab in a solution, and we just do the genome sequence. And we don't actually have a physical isolate virus. And that's very important because an isolate means you have the virus in a tube in a freezer somewhere, not just the genome sequence. The genome sequence is representative of the isolate, but it is not an isolate. So we have hundreds of thousands of genome sequences from SARS-CoV-2, but not that many virus isolates. People get the sequence, they work with the sequence, they don't necessarily work with the virus. A genome sequence represents an isolate, but it is not an isolate. And as I said, there are hundreds of thousands of full genome sequences of SARS-CoV-2, but most are not isolates. The virus was not isolated in cell culture. Now, we give isolates names to keep track of them. I have hundreds of thousands of genome sequences representing isolates or isolates. We want to know a few things about them, like where they came from. So here's some examples. This is a phylogenetic tree of about 4,000 or so genome sequences of SARS-CoV-2 isolates from various people from 2020 through the current time. And you can find this at nextstrain.org. Great resource, by the way. Go over there and just poke around. You can learn a lot by doing that. This phylogenetic tree, what we've done here is to take all the genome sequences and compare them and say which ones are most closely related. And so you can make a phylogenetic tree of those. And you can click on any of these dots. The dots are individual genome sequences, which may or may not have an isolate associated with them. And so here, for example, one of these first isolates back in 2019 is called Wuhan/HU1 2019, Wuhan human-1, and it tells you the date of isolation, December 26, 2019. And the clade number, we'll get to that a bit later. That's not actually the correct way to name these. This shown on the right here is actually how you would name a SARS-CoV-2 isolate. SARS-CoV-2, by the way, is the virus. And then we give the isolates different names so we can keep track of them. Beta coronavirus, it's the genus in which SARS-CoV-2 is located. Wuhan is where it was isolated from the patient, typically an isolate number. Here it's WIV4, which means Wuhan Institute of Virology number 4, and then the year of isolation. You can get a lot of information just from looking at that, right? That's really important. Then down here is another way that we name isolates of influenza viruses. Every virus has their own way of being named in isolates. This was influenza A virus A. There are 3 types, A, B, and C. A/duck. So the second term means what kind of host it came from. The third is the geographical location. The fourth is an isolate number. The fifth is the year, 1968. And then the subtype in terms of the hemagglutinin and neuraminidase, H6N1 in this case. That's great. I love the influenza virus nomenclature. They do a great job telling you an awful lot about the virus. All right, so every virus isolate is named in this way. What about variants? What is a variant now? We've got isolate down. Let's look at variant. A variant is a virus whose genome sequence differs from that of some reference virus. So in the case of SARS-CoV-2, the very first isolate that we had from Wuhan in 2019, everything else that we isolate from then, I'm using isolate in the colloquial sense because we mostly have genome sequences, right? Everything derived from that original would be potentially a variant. So that's what we mean by the genome sequence differs from that of a reference virus. Really important point about variants. No inference is made about whether the change in the genome sequence that you see causes any change in the phenotype of the virus. The phenotype is how the virus behaves. For example, I shouldn't say behave, that's anthropomorphizing, but the properties. For example, the physical stability of the virus, which kind of hosts it can infect, what kind of disease it causes. Those are all phenotypes of the virus. There are many, many different phenotypes, which of course are controlled in part by genotype. So when you say variant, you make no inference about what these changes are doing for the virus. You're just saying, here is a virus that differs in genome sequence from my original isolate. So let's take a look at this now on the context of this next strain.org classification. So again, we have our Wuhan number 1 way back in 2019, which I showed you before. Now I clicked on one of these blue dots downstream of it in the phylogenetic tree.
Now remember, y'all, I showed y'all this months ago, way before our dear brother Vincent Racanelli, right, demonstrated this in March. I kind of stumbled my way up on it, researching some other stuff, and that's how I got to the site. And the next thing you know, it was like a week or so or something later, Vincent had it on the show, and I was like, wow, I can't believe that, you know, I was on the right track. But it all comes from, you know, just kind of knowing. But this is what he's about to do is unique, and it helps explain the variant portion of it more significantly. But as he stated in the beginning of this particular video, that there's this whole word about viruses and strains is unsettled. Most people regurgitate what their teachers teach them. Therefore, we're only repeating what our teachers teach them. But of course, you have some people who want to challenge the narrative to say, no, there is a different strand, is a more purposeful. If it's a new strand, that's a big uh-oh. If it's just a new variant, we understand that because of what I said earlier, natural selection, the ability to mutate and adapt. Let's continue.
And you can see here, it's got an isolate name, Bulgaria 15 2020. That's great. We know it came from Bulgaria. It's given an isolate number 15 in 2020. And in fact, May 13th specifically. And you can see it has these genome changes compared to the Wuhan virus. It has 3 nucleotide mutations from a G to a T at 16,917 base number in the 30,000 base genome. And then amino acid changes, 2 of them, as you can see here. This is not correct to call them amino acid mutations. A mutation is a change in nucleotide sequence. It's very much established. So whoever decided to make this site is wrong about that. I'm sorry. So you can click on any of these dots and see the changes with respect to the ancestral virus. Now, an interesting question is every of these isolates a variant? Because if it has a different genome sequence, it may differ by 1 or 10 or even more nucleotide mutations. Are they variants? I think the answer is yes. The problem is that genome sequencing at this scale is pretty new. And, you know, in the old days meaning before the time we were able to sequence isolates by the hundreds of thousands, variants were more rare because we didn't do much genome sequencing. And so when we found one, we said, yeah, it's a variant. Now almost every one of these isolates on this next strain map is likely a variant. Not every one is different. I could go over some of these dots and you'd see that some of them are identical, but every one of these is different, or nearly every one, so most of them are variants. I'm comfortable with saying that every different SARS-CoV-2 isolate, we're inferring nothing about what those changes do, what those mutations do. Some of them may do nothing, and some of them may have some phenotypic effect on the virus, but I think this should all be called variants. I'm sure you've heard of the term variants of concern, and this arose earlier this year when a new variant was detected in the United Kingdom, and that's described in this technical briefing here, which you can go look up, the title of which is Detection of an Epidemiological Cluster Associated with a New Variant of Concern. So I took this part out for you to see because you can see that they're actually using the nomenclature correctly. And they say nomenclature of variants in the UK. SARS-CoV-2 variants, if considered to have concerning epidemiological, immunological, or pathogenic properties, are raised for formal investigation. At this point, they are designated variant under investigation. That first sentence is really important. These variants, if they have different properties, concerning properties in this case, we investigate them, but we're still calling them variants. If we don't think they have any different properties, we're still going to call them variants. We just won't investigate them. Then they could be designated variant of concern because there's some risk assessment done which says that we're a little worried about these, and that's where that term came from. Now, so they understand that a variant is a virus with a change, as I've told you. And I really appreciate what the UK did, UK scientists did in this case, because they called them variants. And many other people were throwing about the term strain, which, as you'll see in a moment, is not correct to describe these. So they're variants, and that makes it necessary for the press to call them variants because they're variants of concerns. They're not strains of concerns. They're variants of concerns. Okay. Now, what is a strain? A virus strain is a variant that has unique and stable phenotypic characteristics. So you know what phenotype is? It's some property that results from may result from a genetic change, right? And we're saying here that a variant strain is a variant with unique and stable phenotypic characteristics. Unique meaning we've never seen these before. And so the UK scientists call their SARS-CoV-2 viruses isolates variants. They don't call them strains. So when do you call them a strain? Well, that is the key. Not everyone should be able to call it a strain. A journalist describing this should not call it a strain because he or she is not an authority. And you look down here, I put on this slide another quote from that paper I showed earlier, where Jens Kuhn is the first author. The designation of a virus variant as a virus strain would be the responsibility of international expert groups. What does that mean? There are groups charged with virus nomenclature. In fact, the International Committee for the Taxonomy of Viruses, of which Jens Kuhn is a member, decides these issues, what to call viruses. And they would look at all the data at why you think this variant has unique and stable phenotypic characteristics. And then you might call it a strain. But until then, you shouldn't. Why not? It's confusing. If some people call it a strain and others call it a variant, you shouldn't. You should use common nomenclature. And so you see the hierarchy here: isolate, variant, and strain. It's very clear. You do some experiments in the laboratory, in experimental animals, you make observations in people, you put all these together and you say, we have something unique here. We think this merits the appellation strain. As far as I'm concerned, so far, there's one strain of SARS-CoV-2. That could change. For example, last year, a virus with a single amino acid change in the spike, D614G, arose in February, and now pretty much every isolate has that change. So it propagated throughout the world. Clearly has some fitness advantage that allowed it to propagate globally. And when that's figured out, it could get the designation strain. It would be distinct from the original isolate, which is the D at 614, not the G. But it's not been done yet. You shouldn't call it a strain because Jens Kuhn and his colleagues have not said that D614G is part of a strain. Now, you may be asking, what else would make a strain? Well, there are the virus I work with, poliovirus. There are 3 serotypes, which you could call 3 strains because they have unique phenotypic characteristics. They react very differently with antibodies. And by the way, types 2 and 3 have been declared eradicated. So only type 1 is circulating now. So there are these 3 serotypes, and I'll explain serotype in a moment. Then there are mouse-adapted polioviruses. So people have taken type 1 and type 2 and type 3 strains and passaged them in mice, and then they acquire the ability to cause disease in mice. And those are strains because they have unique characteristic, unique being able to reproduce in mice because the human viruses, polioviruses, do not. And they're stable. They don't go away. So there are multiple strains of poliovirus by that criteria. And that's what you would need in order to say that a virus. Variant is a strain, in fact. Now, I mentioned this word serotype. What does that mean? So viruses of the same species, for example, poliovirus, that are antigenically distinct. And I gave an example before, the 3 serotypes of poliovirus. The vaccines that we use to prevent poliomyelitis, there are 2 different kinds of vaccines. There's the Salk vaccine and the Sabin vaccine. They're made very differently. Each of those at one point had 3 serotypes in it. It was a trivalent vaccine because all 3 serotypes were circulating. If you were infected with one, you were protected against it. You never got infected with, say, type 1 again, but you could be infected with types 2 and 3. So they were antigenically distinct, and the antibodies induced by one would not block infection with the other or block disease caused by the other. So that's what a serotype is. It is viruses of the same species that are antigenically distinct. We have 3 serotypes of poliovirus. Now, historically, serotype has been defined by what we call neutralization assays, and I'm showing that to you here on this slide. These are cell culture dishes that have been stained, and these are plaques. These clearings on each one is a plaque, and this is a plaque assay where we can measure virus infectivity. And I have a video which I'll link to above where you can see how a plaque assay works. But basically each clearing or plaque is caused by an infectious virus particle. Now on the bottom, what we've done is we take, I don't know how many plaques are here, 20 plaque forming units. And you can see each of these plates has the same number of plaques because we have mixed these viruses with a saline as a control. On the top, though, we have mixed the viruses with different dilutions of serum taken from an animal that had been infected with poliovirus, say, or from a human that had been immunized with poliovirus vaccine. And here the dilution of 1 to 10, that's the dilution of serum. So it's fairly concentrated. You can see there are no more plaques. You mix the serum with the virus, you incubate it for a short time, then you do the plaque assay. You see there's no infectivity, there are no plaques left. 1 to 100, there are no plaques left. However, at 1 to 1,000 dilution, now you're diluting out the antibodies. You now get 2 plaques. And then at 1 to 10,000, no more neutralizing activity. That's a neutralization assay. And by the way, you would say this serum has a titer of 1 to 100, the last dilution that gave you a blocking of infectivity. So if you do this with type 1 poliovirus, let's say you use type 1 poliovirus as the virus in this experiment, and you have serum from an animal that had been immunized with type 2 poliovirus, you would see no neutralization. It's a different serotype. By looking at the genome sequence, you're actually looking at the genotype, and that's what I want to explain here. It's another term for describing viruses. Genotype, a description of the genetic makeup of the virus. Essentially, do the genome sequence and see if the viruses fall into different genotypes. Now, we do this for some viruses and not others. For example, hepatitis C virus are classified into 6 genotypes. So if you do a genome sequence, you can immediately say this is genotype 1 or 3 or 6, whatever it may be, just by looking at the genome sequence. There's a certain % identity associated with being a genotype. And the hepatitis C viruses are further classified into subtypes like 1A and 1B. So that's genotype, the genetic makeup. And you can look at the genotype now and predict for many viruses the serotype. There's another term you will see called clade. And if you look closely at the nextstrain.org pictures that I've shown you, you see clade on it. And a clade is a group of viruses composed of an ancestor and its descendants. Okay, an ancestor and its descendants. And you can see on this image, I have 4 different phylogenetic trees created from viral genome sequences, say. And in green, that's a clade. All these points or tips of the branches are descended from one ancestor.
Oh, I didn't mean to cut that off, but I'm gonna go ahead and conclude it. I wanted to say something that he had mentioned as far as clades. So in order to better understand that, if we were to take the variants that we have now and compile all of them together, the ancestor to all the new variants that exist now is going to be from China. And then from China it starts to branch out. You get UK. Then you get from the UK you get the South Africa. And then after the South Africa you get India. And then from India you get South America. And then from South America you get the New York variant. From the New York variant you get California variant. From the California variant you get the Colorado variant. So there's 3 different working variants right here in the United States: New York, California, Colorado. They all come back off of what would be South America. I'm walking backwards. South Africa, India, South Africa, UK, and then China. But there could be more, and the United States is behind on tracing those different variants. And that's because a change of power took place. January, we get old Joe in the White House, and then the scientific community get their keys back and access back to certain things that they had control of during the Obama era that they lost control of during the Trump era. So now we're beginning to do more research in areas where we lacked during the previous administration. In short, I'm gonna just conclude this way. Now that we have a clear understanding of what variants are, we can conclude that our news media has totally confused us to the point that we need to rely more on qualified virologists, immunologists, and epidemiologists to fully understand what the science is saying about mutations. If there is a cause for concern, we need to be able to disseminate that information and communicate it effectively to our family members and friends. Here's some sources for y'all. Some more sources. Of course, I'm gonna brand this thing up, and then I'm gonna hit you with the logos, the powerhouse. Now, I do have some additional information that I will cover. Bear with me just a moment. I see we had some nice variant conversations going on in the chat regarding that California variant that people been tooking. I find it very funny though, on the last show when old boy came in and said they smoking on the Zion pack. That was hilarious to me. Yeah, that was hilarious. I think this is one, and I think this is one. And hold on one second. Let me get y'all the right info. Y'all know I got to go to my vault. But I got so much information in there in the middle of writing this book. I got to be careful. I don't want to bring up the right thing. But let me see. I think I sent some stuff to I got some data on reinfection, but that wasn't what I wanted to get into. I'm gonna save that. data. Oh, y'all can see my screen. Let me start sharing my screen. I'm tripping with no map. But those were what you saw wasn't pretty much what I wanted to actually discuss. Let me hit up my other area of where I had data. Appreciate that, Charlie. But yeah, man, what y'all got on them variants? What y'all think about that? That whole little medicine right there. What y'all got? Any questions, comments, concerns? Dang, man, it's too much information. We be talking too much or something. Okay, that was Monday. Let me go back. All right. That's when Floyd knocked the old boy out. Make sure, man, that if you're not subscribed to Science with Sean and you still on the Mossy watching, because there's like there's some people still watching. Make sure that you go over and subscribe to Science with Sean. I want to show y'all this video. Let me see. I wonder if I can I want to do this. I want to show y'all this old video real quick. And then I'm going to do something else. But yeah, man, in short, the reinfection article is pretty much talking about that you have the potential of being reinfected in around 10 months. That's an average, but you could be reinfected before 10 months. You could be reinfected in like 4 months. And you could be infected in 7 months. But the science, the science says what it says, and we just gotta let it continue to do its job. But let me pull this up. Let me pull this up real quick. Y'all gotta see this. I know a lot of y'all probably already saw this, but I'm gonna do it again for people who have not. I should have downloaded it and saved it in my archive. But let me pause it, run it back, close this out, share my screen. Expand it.
See, Sarnetta being nice and shit. The fact of the matter is.
Hold up. So, my boy Craig, 2017, posted, says, Ankh the God Killer of the Master Teacher Amon-Ra Squad goes in on why the conscious community should split. Because around this time, we were just doing certain things different. The Seshu, the Amon-Ra Squad, everything. Like, people were mad at us for challenging information and so on and so forth. So one day, Ankh sent me a text message, said he on Polite channel, right? You know what I'm saying? There's his Polite's building was packed. And on the panel was Brother Reggie, I think Michael Edwards, Inky, Polite, Sarnetta, and somebody else. And this is around the time when Uncle Sarnetta really wasn't talking. and Polite and Sonetta wasn't really talking, so you don't really see Sonetta trying to go in, but he reading comments and stuff. But anyways, he said why pseudoisms is dangerous, and he's saying this in front of Polite, Michael Edwards, Stinky Inky, and Sonetta, specifically the promoters of the pseudoisms, and he was spot on in this. Let me expand out.
See, Sonetta being nice and shit. The fact of the matter is, the damn community need to be split right down the damn middle. You got the pseudos on one end, label that shit properly. You got those dedicated to a method of science and technology that will always uplift black goddamn communities. Plain and simple, bro. All great civilizations at the foundation, what they did was damn STEM. Science, technology, engineering, math, and let's add art to the goddamn equation. These niggas over here, you gotta label that shit. You can never put the pseudos in charge of any goddamn thing. I'm gonna tell you right now, this got better what they did. But these niggas right here, they never succumb to science and technology. They teach their bullshit. So at the end of the day, all of a sudden, they gonna save the fucking community. No, we need to save the community from them. And they lying ass. They got cures from cancer. You know what I'm saying? All that. What about that? Where the hell is the tribunal at that's gonna put them niggas up on charges that claim to have medicine for the community, but they hold it for their damn self? Curing AIDS, curing cancer, and that shit makes some damn shit. So the community need to be split because every place in the world splits science and pseudoisms. Pseudoism, religion over here, science and technology over here, and we good. We need to label that shit what it is. Them niggas don't got no real information, bro. Trust me with that. But see, the first niggas tried and tested because the conscious community is really about information. So now, all of a sudden, the community is splitting up. Damn right we splitting up. All right? At the end of the day, we want the real information that can help us fight the suppression that constantly follows us everywhere. That's what we're doing right now. So I don't care. So check it out. Let's do what all communities do in the world. Put the religion and pseudoisms over here. They label it. You can go to Bob's Nobles right now for like, you got a section that says what? Religion, right? Mythology, right? New age spirituality, technology, don't they? So now we're gonna label that shit. Them niggas teach pseudoism. They got magic in a goddamn bottle, some bullshit, right? All right, pseudoisms, religious, crystals, chakras, all that shit. Put it over here, right? And we still love it. Put the science over here, right? And we do our thing. And that's how we're gonna get along. But I could never join with pseudoism because that's the most dangerous thing that any functioning civilization can have. Pseudoisms is what put the European nations in the fucking dark ages. And not till the Arabs came with the African science that they got from the Greeks that took them out the dark ages. The conscious community is in the dark age.
Man, that was so facts. That was straight facts. It was fire, y'all. I mean, he was, man, listen, he told the truth so many times right there, man. It was sad, man. It was sad. And he did that with the people who promote this shit. Yeah. So I want to read something to y'all real quick out of this biology book. It's not going to, of course, it's not going to be in the book that I'm dealing with, but which is cool. But I want to kind of give it to you the way that, you know, it was received for me. It says microorganisms as a pathogenic agents. It says the 19th century was a period of revolution in scientific thought, particularly in ideas about the origins of living things. The publication of Charles Darwin's The Origin of Species in 1859 crystallized startling and to many people shocking new ideas about the origin of diversity in plants, animals, until then generally attributed directed to the hand of God. These insights, let me say that again because I kind of stumbled. It says the publication of Charles Darwin's The Origin of Species in 1859 crystallized startling and to many people shocking new ideas about the origin of diversity in plants and animals, until then generally attributed directly to the hand of God. These insights permanently undermined the perception that humans were somehow set apart from all other members of the animal kingdom. From the point of view of science of biology, the most important changes were in ideas about the causes of disease. The diversity of microscopic organisms has been appreciated and cataloged since the dawn of recorded human history. However, a vast new world of organisms too small to be visible to the naked eye was revealed through the microscopics of Antony van Leeuwenhoek in 1632 and 1723 is when he died. Van Leeuwenhoek's vivid and exciting description of living microorganisms, the wee animalculus, present in such ordinary materials as rain or seawater, included examples of protozoa, algae, and bacteria. Now, by the early 19th century, the scientific community had accepted the existence of microorganisms and turned to the question of their origin, a topic of fierce debate. Some believe that microorganisms arose spontaneously, for example, in decomposing matter where they were specifically abundant. Others held the view that all were generated by the reproduction as a microscopic organism. The death knell of the spontaneous generation hypothesis was sounded with the famous experiments of Pasteur. He demonstrated that boiled, sterilized, medium remained free of microorganisms as long as it was maintained in special flasks with curved, narrow necks designed to prevent entry of airborne microbes. Pasteur also established that distinct microorganisms were associated with specific processes such as fermentation, an idea that was crucial in development of modern explanation for the causes of disease. From the earliest times, poison air was generally invoked to account for epidemics of contagious disease, and there was little recognition of differences among causative agents. All right, for the people in the back, from the earliest times, poisonous air was generally invoked to account for epidemics of contagious diseases, and there was little recognition of the differences among causative agents. The association of particular microorganisms, initially bacteria, with specific disease can be attributed to the ideas of the German physician Robert Koch. He developed and applied a set of criteria for identification of the agent responsible for a specific disease, a pathogen, articulated in 1890 presentation in Berlin. Now, these criteria that Koch postulates can be summarized as follows. The organism can be regularly associated with disease and its characteristic liaison. The organism must be isolated from the disease host and grow in culture. The disease must reproduce when a pure culture of organism is introduced into a healthy, susceptible host. The same organism must be re-isolated from the experimentally infected host. Now, modern technology has allowed some of Koch's principles to be amended by application of other types of evidence. However, by applying his criteria, Koch demonstrated that anthrax, a common disease of cattle, was caused by a specific bacterium, and that a second distinct bacterial species caused tuberculosis in humans. Guided by these postulates and the methods of sterile culture and isolation of pure preparation of bacteria developed a Pasteur, Lister, and Koch, many pathogenic bacteria were identified and classified during the last part of the 19th century. From these beginnings, investigations into the cause of infectious disease was placed on a secure scientific foundation, the first step toward rational treatment and ultimately control. Furthermore, during the last decade of the 19th century, failures of the paradigm that bacterial and fungal agents are responsible for all diseases led to the identification of a new class of infectious agents, submicroscopic pathogens that can be called viruses. And this is where the discovery of viruses began, in an era where trial and error went through and beliefs got thrown out the window. So to the point of the God of Gaps, in regards to understanding viruses and the emergence of new viruses that became to be known to the rest of mankind, science answered a question that was normally left to be believed by people, that an infectious wind caused certain sicknesses. So shout out to those scientists and their discoveries early on, of their ability to challenge that thought from the late 1600s all the way up into the 19th century. And then we get the discovery of viruses that will emerge. And Ivan Noski, the Russian scientist, was accredited with that discovery. He was able to report of the first pathogenic agent that was smaller than any known bacterium in 1892. And so he got to see the first virus. I found that to be neat. And I just wanted to lay that on people. I know this show is about variants, but we should know that, simply a mutation in something is going to lead to a different part of the original virus being something. So I don't see really any questions in the chat or anything like that. Let me see what y'all got going on. He said it's Sutek drinking Starbucks to stay up. He must be trying to write a paper or something. Yeah, we on okay. I see what Sutek did. Yeah. Some people showed up on the Mossy Hill. Yeah. Somebody asked who the scripture asked what a wine at. But y'all crazy. But yeah, man, pretty much that's where we are. So I don't see any questions regarding to the presentation or anything like that. Yeah, what about Unc? What y'all got for Unc? Oh yeah, I don't know how to order. Well, I can't lie. I did go to Starbucks like once to order a strawberry frappuccino, which I haven't had in a long time. But that's the only thing that I would get from Starbucks is a strawberry frappuccino. I don't drink coffee or nothing like that. So if you ever at Starbucks and I'm in your city, man, you know what I'm saying? I'll take a strawberry frappuccino. They can put the whipped cream on the top. That's cool too. I ain't tripping on that. When I drink it, I ain't gonna get to, I ain't gonna drink the whipped cream part of it or eat the whipped cream, but for presentation's sake, it look good. Do they even make the strawberry frappuccino anymore? Because I used to be able to buy them. I used to be able to buy them in the store. Like I would go to the store, and you know how they say the little 4-pack. Yeah, I used to buy them jokers, man. Store them in the refrigerator and then just pop one out. I used to go like twice a week to get them jokers when I was hustling. Yep. All right, let me drop something else on y'all, man, since y'all want to go there. Intracellular parasitisms of viruses. Now, you're going to hear me say something about intracellular parasites in the up-and-coming book. So I'm not going to give you that definition, which I can expound on in my book right here. But I want to go down to a point where it says the transmission of yellow fever virus to mice by Max Thieler in 1930 was an achievement that led to the isolation of a tuinated strain, considered one of the safest and most still considered one of the safest and most effective ever produced for the vaccination of humans. So look up Max Thieler, 1930. He was able to, you know what I mean, develop a way to handle yellow fever. After specific viruses and appropriated host organisms were identified, it became possible to produce sufficient quantities of virus particles for study of their physical and chemical properties and the consequences of the infection for the host. Features such as the incubation period, symptoms of infection, and effects of specific tissues and organisms were investigated. Laboratory animals remain an essential tool in the investigation of the pathogenesis of viruses that cause disease. However, real progress toward understanding the mechanism of virus reproduction was made only with the development of cell culture systems. The first and the simplest, but crucial to both virology and molecular biology, were cultures of bacterial cells. So think about where we are today. Yep. Think about where we are today. That's a big deal, man. Y'all don't see it, but I see it. I might be geeked out and all that, but it's all good over here. Transmission of plant viruses such as tobacco mosaic virus can be achieved readily, for example, by applying extracts of an infected plant to a scratch made on the leaf of a healthy plant. I'm going to say that to y'all again. Listen to this. Transmission of a plant virus such as tobacco mosaic virus can be achieved readily, for example, by applying extracts of an infected plant to a scratch made on a leaf of a healthy plant. So if I have two plants, one's infected, the other one is not, I can just scratch the other plant and I can place the infected plant on the other plant and then the healthy plant, and then the healthy plant is sick. Think about yourselves in that matter. Think about how we encounter viruses daily. Remember that 6 liters of oxygen you breathe, how many viruses are contained within that 6 liters? But this is why they study plant viruses, because of that example that I just gave you. Yep. I don't even want to go back and add that in that work, man. It's so much. Like, I ain't even putting a whole lot of stuff in there in this book. I'm just trying to give people like the basics so that they can have a solid foundation when actually trying to understand what the science is saying. I can't wait to just see how people are, in my opinion, writing it and all that. I don't think I'm doing a really good job. So when y'all get it, man, I just really need your honest opinion on what y'all think about the actual book. What y'all know about mRNA? Not a vaccine, but just mRNA. What y'all think about mRNA? Everybody think about mRNA, they think about vaccines. But what about mRNA answering questions about our existence, y'all think about that? I'm gonna see if I can find this article that I was looking for. And let's have a little bit of talk about biology in regards to mRNA. Dang, I'm an hour and a half in. Nah, I ain't talking about vaccines, bro. I'm talking about life form. See, I ain't talking about vaccines. Look at this. So look, I'm on Khan Academy, right? Y'all see it. Look what it says over here, the origins of life, right? The origins of life. And there's a transcript. Now, I already read the thing, but let's see where this conversation goes. Oh, this video 10 minutes. I ain't gonna do that. RNA, origin of life. Let's go. This video 3 minutes.
Where does life come from? This is one of the most important questions humanity has ever posed, and the scientific answer is, we don't entirely know. You might think that cracking DNA's genetic code should have explained life's origins, and it definitely helped. Thanks to our understanding of DNA, we can map out the history of evolution all the way back to single-celled life. But that's where we're stuck. The problem is, DNA is a great way to store information, but it doesn't do much else. Cells rely on other molecules like proteins to replicate, grow, and survive. Proteins, on the other hand, work great as molecular machines to keep cells alive and healthy, but they can't store information or copy themselves. They need DNA for that. So we have a chicken and egg problem. DNA needs proteins to function, and proteins need DNA to exist. So which came first? Which molecule made life possible? Well, there's a third type of molecule that may hold the answer: RNA. Most scientists think that RNA came first because RNA can do two jobs: store information and perform various functions that keep cells alive. This idea that RNA came first is called the RNA world hypothesis. RNA world suggests that billions of years ago, in some primordial soup of molecules, a self-replicating RNA formed. This may have happened in volcanic vents deep on the ocean floor, or perhaps clay clumps brought the necessary chemical building blocks together. Some scientists have even speculated that early RNAs formed on Mars and hitched a ride on an asteroid to our planet. One way or another, self-replicating RNAs emerged, multiplied, and evolved. Over millions of years, they developed into a legion of molecular machines. These microscopic proto-life forms blossomed and competed. The best collections of code lived on, and the weaker ones died out. Survival of the fittest was the name of the game. This competition for survival eventually led RNAs to evolve the ability to build strong, stable proteins, which excelled at carrying out complex biological processes. And somewhere along the line, some critical RNAs mutated into the familiar double helix of DNA. DNA became a stable archive of genetic information that stored blueprints for the most successful RNA and protein molecules. Life became more complex over trillions of tiny steps and happy accidents. And all the while, the RNA lineup grew alongside lengthening genomes of DNA and complex proteins. And it's all still happening inside your body. RNAs have adapted to become the Swiss Army knives of our cells. Today, they can slice, dice, catalyze, build, destroy, code, replicate, and transform. A remarkable diversity from the simplest of beginnings, a single self-replicating RNA molecule.
Man, this thing, it's gonna get deep. RNA world hypothesis. First on RNA is like, it's pretty much what that video was right here. So it's just talking about some of this stuff. But see, look, y'all see that mRNA in there? So let me get to the point. Now, this ain't talking about no vaccine. See what I'm saying? We're talking evolution. We ain't talking vaccine, right? So when we think mRNA, we're talking about messenger RNA, we're talking about vaccines. But watch this. Some messengers, RNAs, mRNAs, and bacteria and some plants contain a section of code called a ribose switch that can grab onto a specific molecule. Binding this molecule controls whether the mRNA is translated to make a protein. The molecule could be a nutrient that binds to an mRNA ribose switch and triggers the mRNA to translate to make a protein that breaks this nutrient down. So mRNAs that contain ribose switches can regulate themselves in response to specific molecules. It was previously thought that only proteins regulated the production of protein from mRNA. However, ribose switches hint of a regulation system that may have existed in an RNA world long before proteins existed. Yeah. Yep. Look, one messenger RNA can be remixed in different ways so that its genetic code can be translated to make lots of different proteins. The ability to make more than one protein from one mRNA sped up the evolution of multicellular life. Newly made mRNA is spliced by a molecular machine called the spliceosome that is like cells and glue of the cell, made of RNAs and proteins. This machine chops out unwanted sections of mRNA code and sticks the remaining mRNA back together again to create mature mRNA that can be translated to make proteins. Now, back in the 70s, scientists thought that one gene coded for one mRNA, which in turn coded for one protein. This is largely true of the bacteria and other single cell life. However, for multicellular life, one gene codes for one mRNA that can be spliced in different ways to create many different proteins. This is called alternative splicing. Alternative splicing is an ingenious way of creating a diverse range of proteins from relatively small number of genes. One of the surprises of the Human Genome Project was that the human genome codes for so few genes. Scientists predicted that there would be about 100,000 human genes, but the number is closer to 20,000. These genes are spliced in different ways to create a huge number of human proteins. Speeding up evolution. The arrival of alternative splicing in multicellular life forms likely sped up evolution. It meant that an organism could create new proteins without going through the lengthy process of evolving new genes. Chance mutation would have arising that caused existing mRNAs to be spliced in different ways. These alternative spliced mRNAs coded for brand new proteins that may have driven brand new cellular processes, driving forward the evolution of complex life. It's too much, I know. It's too much. You can't fathom that. I know you can't. I understand. You just thought mRNA had something to do with just a vaccine. I understand. I didn't mean to drop something on y'all that y'all wasn't aware of. I apologize. This is just some of the stuff that, you just run across from time to time and have conversations about, with your people and things of that nature. Forgive me. That's correct, Zane. It is correct. True Tech over there talking about magic. Yeah, man. So look, man, I really appreciate everybody sliding through. We've been on for about an hour 40. I was gonna try to drag it to an hour 45. but you know what I'm saying? I know it's, it's Thursday. Everybody got to work tomorrow, and then you have your weekend and so forth. So uh, I really appreciate everybody for tuning in. Thank you for your time and patience. If you haven't subscribed to Science with Sean, please subscribe to Science with Sean YouTube channel. We are going to this weekend. Hopefully, I may be able to present again before I get into chapter 4 of my latest book that I began to work on. The chapters are, as I go through these chapters, they will get easier for me to write. The first 3 chapters were very challenging from a scientific perspective because I'm talking about some things that I'm learning in that process. And in return, I'm trying to demonstrate what I'm learning by using examples, sources, definitions, all kinds of things that I could source. Like in chapter 1, it's already like 21, 22 sources in chapter 1 alone by itself. You know what I'm saying? So that's a lot of sources, a lot of information compacted in chapter 1 to set a foundation and then to build on. And some chapters are condensed sources out of the first few that I've written, and some are comparable. However, the goal is to get the book in your hands as quickly as possible so that you can learn what I learned. Pretty much not only learn what I learned, but there might be some understanding. So like a YouTube video may not be able to expound on or something like that. So hopefully the work is appreciated, and I don't expect tons of people to actually buy the book or anything like that. But if people grab the book, support the book, and suggest the book, then thank you for that. However, Sutek said it earlier that this is going to be another good year for the Kofi Pi-Psi research team because of individual projects and then the collective projects. So hopefully, we continue as a team to properly educate the masses and stand on scholarship the way that our ancestors did before this era of ignorance came in. So on that note, man, I already know how I'm rocking and rolling. It's Shimon Hotel, and
Log in to KofiPiSciTV.com and get Spears of the Mossy Volume 1 by Kofi Pi-Psi Research Team, from Spears the Pen Volume 2 by Kofi Pi-Psi Research Team, Spiritual Combat Volume 3 by Kofi Pi-Psi Research Team, One Night Before Bed by Any Harret Sean Calfunny, Kalawa's Trip to the Motherland by Kofi Pi-Psi, From Cocaine to Consciousness by Any Harret Sean Calfunny, As I Learned, We All Learn by Kofi Pi-Psi. Also, if you want fresh merch, go to our online store, Family Connect Apparel, and get all the brands. Also, get our 54 deck of quiz and educational cards available now on KofiPiSciTV.com.